we're going to wait. Right, I shall click on them. Do you want me to get, actually, I need to share my screen, don't I? There. Is that nice and legible?
Yes.
Shall I get going?
Okay, all right, we'll get started. People
Get started, right? Okay, so we're talking about oxygen. And one of the difficulties I find in reading various papers is actually people use the same term to mean different things. And there's not necessarily that much consistent, but everyone's aware of air pressure and that it goes down as you go up. And if you measure it in newtons per square meter, it's 1.013 times 10 to the power 5 newtons per square meter. People are more used to millimeters of mercury. A bar is ordinary pressure, and an atmosphere is an ordinary pressure, which is like the 76 centimeters or 760 millimeters of mercury. When you look at the way in which different gases respond, you need to think about the partial pressure. That is the part of the total pressure that relates to one gas. And because we're talking about oxygen, this will be oxygen, but it could be anything else. And it obviously affects the solution in liquid. Now, liquid here is water. It's in blood serum. And it's the partial pressure. And then we have the terms hyperoxia, higher oxygen, hypoxia, low oxygen, normoxia, hyperbaric, high pressure, normobaric and hyperbaric, normal pressure and low pressure, from which we get the normobaric oxygen paradox, which is a very interesting thing that I shall refer to. A couple of the papers I linked to talk about the normobaric oxygen paradox. Not necessarily here, but on one of the other pages. But moving on. So, and I'm just going to go through these quite quickly. People, I'm sure, know these things to some extent. That the normal atmosphere is about a fifth oxygen. And about sort of like 78% nitrogen. Oddly enough, almost 1% argon, and then there's some carbon dioxide and varying amounts of water. And what I found interesting in some of the papers is that we started out with oxygen being a higher percentage in air. I don't know how they worked that one out, but it was a long time ago. It was 40% when there was life. And if you say the normal pressure of oxygen, a bar of partial pressure of oxygen, which is 21%, then that's 160 millimeters of mercury. That is normal partial pressure of oxygen. And it has solubility in water of oxygen, nitrogen, carbon dioxide. So the interesting thing about that, and we could go on to the last thing on here, is they did some experiments. I couldn't find a reference to this, so I haven't put a reference in, but they actually kept a pig alive in high pressure with a high partial pressure of oxygen, but without any blood, without any hemoglobin, because you could get 6% oxygen in water, and that was then possible. But the thing about passing oxygen through the body is that it obviously starts in the lungs at whatever partial pressure you've got. So we're talking about 160 millimeters of partial pressure in the lung. For it to come out of hemoglobin into the serum, you need about 20 to 40 millimeters of mercury. And then the last step is just oxygen dissolved in the serum. And so when you increase or reduce the partial pressure of oxygen in the lungs, that has an effect throughout the body. And the last step is governed by that to a certain extent. So moving on again.
Well, that's interesting. They kept the pig alive without hemoglobin by increasing the percentage.
I think it was three atmospheres of pressure with a high concentration.
So the oxygen just diffused. It didn't need the bloodstream.
Well, it dissolved in. They had got 6% of oxygen in blood, in water, and so you didn't need hemoglobin to get it to the tissues because there was so much in the water.
And did it live a normal life course? Do you know?
Well, I don't think they did it for a long time.
Interesting.
I'm sorry, I couldn't find. I don't know whether it's. I've got this nice book here, which is Handbook of Hyperbaric Oxygen Therapy, which is a very good book. I did tell you what the book was in the Discord, and it's a very good book if you want to look more at all of this, because there's a lot of really interesting stuff. But yeah, you don't your hemoglobin is there to get a volume of oxygen through, but actually the cells don't really want a very high level of oxygen anyway. All right, and these are quotes from one of the papers I've linked through, which show your sort of obviously the oxygen tension, the oxygen concentration in the liquid goes down and down and down as it gets to the tissues, and it goes down as it comes through the cell and goes to the mitochondria. So this is quite a good intro here, but I'm not going to read it out. But basically, there was like 60 millimeters of mercury in the bladder in a rat, muscle 40 millimeters, liver 20 millimeters, renal cortex, etc. All right, and obviously the level of oxygen depends on how much oxygen you're consuming. Because if you're not using very much oxygen, it's not going to go down that much. It'll average out. So the last step is to mitochondria, and it passes in both the water of the blood serum and hemoglobin. And high enough pressure can obviate the need for hemoglobin, but we don't really want to do that. There are two ways you can increase partial pressure of oxygen. One of them is to have a higher proportion of oxygen, so you can have like air at room sea level pressure, but with say 40% oxygen, or you can take ordinary air and double the pressure. And in fact, you can do both. So very often when people do hyperbaric oxygen therapy, they will both increase the pressure, but also increase the proportion of oxygen by using an oxygen concentrator. So people breathe air from an oxygen concentrator at a higher pressure. So say the oxygen concentrator takes it up to 40%, to say 1.4%, then you increase it in two different ways. Now, I think people know that hypoxia, sustained hypoxia, low oxygen, is toxic. Do people know that? I presume they do. You've heard about brain damage from people not having enough oxygen when they're born. All right.
Yes.
But also sustained hyperoxygen is also toxic. So when you have a high level of oxygen, it generates more ROS. And obviously people know that in the mountains, as you go up, the pressure goes down. The proportion of oxygen remains the same, roughly, but the pressure goes down. So the partial pressure of goes down. So Everest, which is about a third of sea level pressure, has a partial pressure about 55 millimeters of mercury. And similarly, as we go under the sea, we've got sea level pressure is the normal bar. As you go under the sea, about 10 meters of water gives you 1 atmosphere. And so people who are scuba divers, they're not trying to have a partial pressure of oxygen greater than 1.5 times the sea level pressure. That's the 160 millimeters of mercury, because of its long-term toxicity. And like below 197 feet, they try and have a special air mixture, which is 10% oxygen, so that the partial, once you increase the pressure on that, the partial pressure isn't so high that it's causing damage to the person breathing it. So you know all of that. Now I got this table, which is an interesting table to look at, just to see the various partial pressures. So atmospheric, we know that at around 2.5 kilometers, 2,500 meters in the mountains, people can live. It's sometimes hard to have babies there, but you can live in that environment, and that's with a partial pressure, not of 160 or 159 millimeters of mercury, but 125 millimeters of mercury. All right. So it's interesting when you think about it. You've got sort of like, as you go up, there's a problem, and as you go down, there's a problem. So if you go down below the sea, there's a problem. And I don't want to go into why there is a problem so much, although Ross is part of it. But going on, we also know that the naked mole rats, which are a non-Gompertz creature, that is, their mortality does not increase exponentially. they live in a hypoxic environment. And the bowhead whales that live 200 years also live in a hypoxic environment. Now, here's a question for people. When did you, as an individual, last experience serious hypoxia and survive? And how low was the partial pressure of oxygen? Does anyone want to guess? No guesses?
Like swimming?
Right. Well, actually, when you were conceived, because the mammalian reproductive tract is supposed to have a partial pressure of only about 40 millimeters of mercury. So that's much less than Everest. Well, I say much, it's less than Everest. Now, obviously, there is a question as to what the partial pressure is in the egg itself. But there is this interesting thing that when people are conceived, they're conceived in a hypoxic environment. And this also feeds into the interplay between hypoxia and pluripotency.
There's a drastic difference between the hypoxic environment of bowhead whales and naked mole rats. Is that right?
I'm not, well, I don't know exactly. Obviously, the naked mole rats are in tunnels, all right? And this hypoxia is not going to be as low as 40 millimeters of mercury. Bowhead whales, they're in a hypoxic environment. I'm going to come on to the hypoxia inducible factor, which is what this is mainly about soon. But I just want to point out this paper here, which I'm not going to go into in great detail, but it basically says that at the point at which people are conceived, they're conceived in a very low oxygen environment, all right? And that suggesting that the oxygen levels influence the factors that maintain pluripotency, all right? Stem cell niches are hypoxic, all right? So this is just another paper. I've got a second one on stem cell niches being hypoxic, all right? So then I move on to low oxygen levels, switch off oxidative phosphorylation. We know about how it can go towards lactate, right? So that you switch off oxphos and you start fermenting. But actually, there is the non-canonical TCA cycle. Now, I'm going to go to this because this is an extract from the paper which I just linked to. And everyone, I'm sure, is aware of the tricarboxylate or Krebs or citric acid cycle, and the traditional citric acid cycle that goes on in the mitochondria. But we also have a non-canonical TCA cycle. And this is interesting. If you see the hashed line on here, the dotted line, that's the mitochondrial membrane. So pyruvate comes into the mitochondria, gets converted to acetyl-CoA. That then is linked with oxaloacetate to create citrate. And then that is swapped with malate in the citrate carrier, right, which is SLC25A1. And then that goes into the cytosol, gets converted back to oxaloacetate, that gets converted to malate, which then swaps with citrate and goes back into the TCA, the mitochondria. That's converted to oxaloacetate, and it goes round and round and round, right? And what's interesting about this is its role in maintaining stem cells and pluripotency. And the point about this is that the acetyl-CoA that is in the cytosol is then available for ACLY, well, it's ACLY is created. It's then available to go into the nucleus and be used to acetylate the histone. So it opens up the histone for transcription. And there is this really interesting question, which I haven't been able to get answers. There's only questions in the literature. There's no answer on this. But there is a point at which the acetylation could be non-enzymatic. So you get a much greater level of acetylation in the histone. And that you know, this is a really interesting area, which is this sort of whole question of how the differentiation of cells is linked to these processes. And the role of SLC25A1, which is interestingly identified as being causative in terms of the failure of differentiation of osteoblasts rather. All right, this is the whole point about osteoporosis, where the osteoclasts are created, but the osteoblasts are not created. It is this failure of the non-canonical TCA cycle through a shortage, they say in the paper that I referred to the last time I did this, of SLC25A1, all right, which is the citrate carrier. Okay, so that's this paper here, which is a very good paper, and I do recommend reading through this paper, which goes, but this sort of collectively, these studies underscore the diversity of metabolic strategies that support cellular bioenergetics and reveal the TCA cycle behavior is dynamic and entwined with cell states. So this is this whole differentiation linked to a different approach, which is a cycle. So you don't end up with lactate. You end up with pyruvate going into the mitochondria and then acetyl-CoA being available in the cytosol as a consequence of that process. So, and there's an interesting question. Professor Thomas Seyfried has this metabolic theory of cancer, which is also linked to moving towards fermentation rather than OXPHOS. Right? But I just passed that one over quickly. Now, okay, this is where I really think we start getting into interesting questions about pragmatic things that one can do to improve longevity, and perhaps why the naked mole rats and the bowhead whales have such long lifespans and health spans. There are 3 key cellular transcription factors that are relevant to questions of oxygen. There's the hyperoxia inducible factor 1 alpha, the nuclear factor erythroid 2 related factor 2, and the nuclear factor kappa light chain enhancer of activated B cells, NF-kappa B, which is very rarely referred to as the light chain enhancer of activated B cells. And I like to think of these as being 3 different responses to damage in a cell. And HIF1 alpha are the builders. When you stimulate HIF1 alpha, it builds new things. And when you stimulate NF or NRF2, it's the firemen. They generate more antioxidants. And when you go as far as NF-kappa B, you bring in the army to have a fight. And in fact, NF-kappa B will increase the citrate carrier and increase cytosolic ROS, so that the individual cell can then fight other cells or bacteria or whatever it wants to fight in the area. Now, I personally think there's a reasonable chance that the hormesis, the U-shaped curve that you see from hormesis is linked to this in some way, or at least there are aspects of commonality, that the sort of a little bit of stress gives you HIF, a bit more stress gives you NRF2, and a lot of stress gives you NF-kappa B. All right. Now, this is where I want to look at the normobaric oxygen paradox, and we can look at that in much more detail when it comes to the actual papers that look specifically at that. Interestingly, I think everyone knows that hyperbolic oxygen therapy is known to have beneficial effects on things like telomeres and various other things. And there are questions as to what extent that arises from stimulating HIF and to what extent it arises from other things. And there's probably multiple effects. But what is really interesting about HIF is if you take the oxygen partial pressure up and then drop it back to normal levels, you actually start stimulating HIF1 alpha. And in fact, you can do that at normal air pressures, so at sea level pressures, just by increasing the concentration of oxygen in the normal air. Right? So it doesn't necessarily require increasing the actual air pressure. It only requires increasing the partial pressure. And that's really interesting from a therapeutic point of view. An interesting thing as well is that exercise also stimulates HIF. Again, HIF is a builder. It says we want more stem cells. It says we want to generate potentially angiogenesis. And there are problems with cancer, because when HIF is stimulated with cancer, you can get the blood cells to go into the tumor to enable the tumor to grow. So it's not always positive. So what is the challenge? It's getting the builders to work on the house without setting it on fire or starting a war. And how can we use oxygen to achieve this? Now, I'm now going to go to my browser. So I shall stop sharing this, and I will go to share my browser, where I have linked to loads of, this is the last thing, which is on my blog, where I've linked to loads of papers which look at HIF, NRF2, and when I wrote this was a few years ago. Yeah, have we got that there? Yes, we've got that there. Right, okay. I tried to find all the papers I could that were about stimulating responses with oxygen. So, and here we have, obviously, you've got the link to this in the chat. The normobaric oxygen paradox. Here, a new tool for the anaesthetist. That's one of the paid papers. That's free to air. You can get hold of the PDF on that, and that refers to how you can get a HIF response by taking the partial pressure up and then dropping it back, but actually without going any higher than normal. Some people, what they do with this is they do exercise wearing an oxygen concentrator. My own personal view, and I'll show you the oxygen concentrator in a moment, is you don't need to be doing such a complicated thing, that you can actually do it just with an oxygen concentrator without the exercise, which is obviously a lot less hassle. So, but what I did was I looked at all these papers, and I analyzed the paper looking at how long you were exposing somebody to a high level of high concentration of oxygen. And there was a particular study on the normobaric oxygen paradox, which I've linked to that one here, and I'll open that one up very quickly. All right. And this is a really good study, because this is looking at the different responses to oxygen stress.
Can you please summarize what the paradox is?
The paradox is that you get a hypoxic result from hyperoxia. That's the paradox. The paradox is that by increasing the partial pressure of oxygen in breathed air, you get a response which is a hypoxic response, which you can also get with hypoxia. But obviously, you can get the hypoxic response by cutting a lot of oxygen out of the air you breathe. Well, there's a problem with that, which is you get brain damage, right? But if you take the partial pressure of oxygen up for a short period of time, the question is how long, and then drop it back. And here they looked here at 3 oxygen levels, 30% of normal, 100% of normal, and 140% of normal. Now, to get 140%, obviously you're using hyperbaric pressures as well, because you can't get 140% without doing that. But you can see here that the HIF response occurred actually on the relatively low focuses. And looking at the other papers here, which didn't always look at HIF, some only looked at NRF2, and some, they looked in various things. But what it demonstrates to some extent is that the HIF response is a transitive response. It responds to that change coming back down. So you need to create a stability at a higher partial pressure of oxygen, which is affecting how the mitochondria are generating ROS. But then you drop it back, Now I'll just show you. I don't know how many of you have you seen an oxygen concentrator? I shall take the camera, and I'll stop sharing just a sec. Well, we can talk a bit. I can always go back to sharing. That blue thing there is an oxygen concentrator.
Ah,
Now,
wait,
if
what
I
do you?
switch it on, right, you can get this is the mask. I'll just put the mask on. All right, now I'm wearing the mask. And we put the oxygen concentrator on. And it's all right, I'll switch it on. Okay, because obviously it makes it hard for me to talk with the mask. But that's an oxygen concentrator. And that people use those in hyperbaric oxygen tents or when they do dives. Now that generates 95% oxygen, but obviously it gets mixed with air. And you might only get 60% oxygen in the breathed air itself. But what it does indicate that at least in theory, if you breathe that for a period of time, and the experimental results indicate that, then you can stimulate HIF. Now, obviously, it's much easier than going and sitting in a tent and turning the pressure up to 1.4 or 1.8 bars.
Wait, do you have an oxygen tank attached to that, or what is that machine?
No, it's an oxygen concentrator.
So it's taking it in from the surrounding.
It takes the air at 21% oxygen, and it concentrates it to 95% oxygen. Right, they cost £300 sterling, which is probably about $350, something like that. They're not particularly expensive. Whereas if you want to go and buy a hyperbaric tent, it might cost you 20,000, something like that. But the interesting question from it, obviously, it's supposed to encourage the creation of new red blood cells, angiogenesis. I think it probably stimulates stem cells. There's really good evidence that it stimulates the build part of the cycle in the body to stimulate potentially extra stem cells. We don't know really, do we? But it's an interesting therapy question as to, we know it works because it's been replicated by a number of people. There you go. It's an interesting thought, isn't it? I'm very happy to go into more detail in any of this. I just thought if I just run through it reasonably quickly.
You're saying that there's a lot of evidence for the greater presence of oxygen will have beneficial effects, such as anti-aging effects, such as increased stem cell activity. So why hasn't so our body has the ability to do all these beneficial effects in response to increased oxygen presence. Why doesn't it?
It's in response to a reduction of increase. It's the change back to normal. It's not keeping it high.
Oh, sorry, I didn't understand that. So
yeah, it's the normobaric oxygen paradox. It's actually that when you reduce the concentration, the partial pressure of oxygen, it generates the HIF transcription factor.
So you increase the pressure, and that's not what's causing the beneficial effects. It's then going back to normal. That's when you think you get the effect.
That's when you get it. That is what's called the normobaric oxygen paradox. And
and the beneficial effects are because it activates beneficial pathways in the body. Why weren't those pathways already activated?
Well, they won't be activated because they're only activated for hypoxia. Yeah, but they are activated by other ways of achieving. Exercise is another way of increasing. Exercise just in normal atmospheric oxygen can stimulate HIF. Now, the question we don't know the answer to is exactly what levels of effects it has in doing this. Obviously, there's been experimentation done with hyperbaric oxygen, and very often that's done with 20-minute breaks. And I think that we know that the 20-minute break itself can be the thing that stimulates the HIF response. What we don't know is how long you need to keep the partial pressure up for. I would think it's minutes. I wouldn't think it's seconds. But the experiments were done like with 20 minutes and 30 minutes, which isn't a long period of time. But what we do know is that if you keep a high level of oxygen for a long period of time, it does damage. And obviously, it's generating more ROS because it's overclocking the mitochondria a little bit.
Yeah, very interesting. I'm still, can you talk more about how this fits into your theory of aging and why evolution, how it fits in
Well,
evolutionarily?
I don't. Well, this is not. This is something which it isn't necessarily doesn't have to fit with the theory of aging. Do you see what I mean? As in, they're two separate things. This is a transcription factor. These are three transcription factors where there is quite a lot of research. And I've done a web page which is linked to all of the. I can bring the web page up and we can go through each individual paper if you'd like. Do you want to do that?
Yeah, we can. Yeah, but very must have taken a lot of
I
time.
don't mind. I'm in no rush. I can spend quite a lot of time going through all of this.
Wait, but just
Um,
first explain how it fits in. It does. You think it's related to aging, right? You think perhaps.
I think no. I think it's a mechanism you can use to stimulate positive responses. So we know there are some interesting things we know. Exercise is important. Okay. We know actually that if you exercise one arm with the other arm in a cast, the other arm will grow, and that the
Oh,
body has.
really?
Yes, we do know that.
The arm and
Wow.
the cast will grow? Oh, yeah, I didn't know
That's
that.
right. Yes, we do know that.
That's
I can try and find that. If I try and find it now, it'll probably take me a bit of time. But
If you put a
it's not.
wait.
cast on the arm, the muscle will destroy itself, but you can
Yes, I
mitigate
know. But
that.
if you'd exercise the other arm, you see what
Well,
it is that there is an effect of exercise on the whole body. So
wait,
if you
so do they both get the same benefits of the
no, they
exercise?
don't both get the same
Okay.
benefits. No,
I
it's
was
not.
hoping I could cut my
Exercise.
workout in half.
Sorry.
I was hoping I could cut my workout in half, like
Well, you
only
can't cut
exercise.
your workout in half, no, because but what it is there is a whole body benefit to exercise. I don't know what there must be some form of cytokine or something trundling around with messaging to encourage things around the body. And obviously, you've got the extracellular vesicles and the transfer of mitochondria and all of this.
It's similar to what I'm trying to say before. Whatever your body does in response to exercise, it could have done in response to low activity. It doesn't. It's not about the fact that I'm necessarily tearing muscle fibers or cracking bones. It's about the response. Exercise activates a lot of pathways that have anti-aging effects. Yeah, that's
That's
right.
right.
On an organismal level.
And I
And
think one of them is HIP. Well, I know
yeah,
one of them is HIF, and I think HIF itself has this benefit.
yeah.
And you can also stimulate it through the normobaric oxygen paradox, which is quite a good thing.
So
Uh,
evolution is telling us that the weak don't deserve to live long.
I think it's use it or lose it as well. That
Hmm.
if you are not using part of your anatomy, it
But
starts
why?
fading.
Why is it use it or lose it? Why do I have to work out hard and I have to change the oxygen concentrations in order to activate these pathways? Why hasn't it just
Changing the
selected?
changing the oxygen concentrations is a bit of a shortcut. It's not necessarily what it's designed for. I think when you consider evolution has really tried to design bodies to cope with varying amounts of nutrition. And so you don't necessarily want to maintain function that is not being used because it's going through a process of renewal all the time. And there is an energy and nutrient cost to maintaining things that are not being used. And if you've got limits on nutrition, you want to maintain the things. So if you are
There's a cost for me as an individual or for the
well
group?
for the individual, yes, because obviously if you've got somebody like in a wheelchair but they exercise their arms become stronger and their legs become weaker. Yeah.
Yeah, well, there is a horrible cost to the individual of obesity, right? So evolution, why has it done that?
Yes, because evolution generally has an environment in which there isn't that much food available. So the actual over availability of the food.
Yeah, I could show you papers where we observe animals in the wild and obesity happens in the wild.
It will do if there's enough food, yes. But evolution, I would not argue evolution is perfect. I would just argue that it selects more food to stop species dying off from a shortage of food, because you will have famines from time to time. And you will not want to maintain unnecessary facilities. You'll want to reduce the energy costs.
Yeah, I mean, this is actually kind of the direction I want you to go in. Can you explain maybe evolutionarily? All right, so you change the concentration of oxygen, it has anti-aging effects. What does that imply for our theories of aging?
I don't think it necessarily implies anything. I think it's interesting as a tool for getting at the control levers in the cell. So it's a tool for impacting on HIF throughout the body, rather than just in these. You see, if you're doing exercise, you are creating localized hypoxia to a certain extent. But obviously,
you don't think it has anything to do with aging because you don't think evolution was just responding. There's nothing.
I think the fact that you can do this by varying partial pressure of oxygen is just, that's the way it works. Because if you look at the creatures that have long lives that live in hypoxic environments, if you don't mind, I'll switch my phone off. There we go. If you look at the creatures that live in hypoxic environments, viz the naked rollout and the bowhead whale, they're benefiting, I would argue, potentially from exercising this particular pathway more often than other creatures exercise it. All creatures exercise it pretty well.
So we have an evolutionarily conserved pathway in which changing oxygen concentration leads to life or anti-aging effects, maybe life
It leads
extension.
to a building effect. So it's it's an anabolic effect, really.
A building effect in the hypoxic environments, and they exercise this pathway more.
I think it's hormesis more generally, actually.
So you think if we took a naked mole rat and we can keep them, I mean, it's very.
Well, I don't know what they do with naked mole rats. In fact, they've lasted quite a while. I don't
let's
know
say
if
we took the
they
naked
keep them
mole
in their
rat.
tunnels. Do they keep
Yeah.
them in their natural environment, which is hypoxic tunnels, or do they actually keep them in a sort of like a rat cage?
Yeah, that's what I'm saying. In
I
the
don't know is the answer. I've tried to find out, but I haven't found out. You could
Okay.
try and find out. It'd be an interesting question to what extent the naked mole rat's extended lifespan or non-Gompert's lifespan is affected by hypoxia. That would be a very interesting question.
Yeah, you
It
said
doesn't in itself stop the issue of hypoxia being an interesting issue, because that the interplay between hypoxia and stem cell differentiation is an interesting question as well, because it's very clear that you, by maintaining the higher levels, well, this is the whole question of what happens when cells differentiate. What happens to stop them differentiating properly? All of these really interesting questions.
great.
And
It's
so
great.
hypoxia has something to do with stem cell differentiation?
Yes, it does. I could go back to those papers. I went through. I don't know whether you were on the call at the time when I went through those papers.
No, I was late today, so yeah, I think I missed that part.
Yeah, I've put in a link in the chat to all the papers, and there were two of them referring to stem cell niches being hypoxic. And the thing about shall I go back to the non-canonical TCA cycle? Shall I do that? Yeah.
Yes.
Yeah, I'm curious about it.
Yeah, let's go back to the non-canonical TCA cycle. So I've got to get.
You were saying this non-canonical cycle is activated in response to a return to normal concentration?
No, no, this is a slightly different thing. I think it's what we're seeing is a lot of things pointing out the importance of hypoxia. All right. What we haven't got is all the jigsaw put together with all the pieces in the right place. All right. So we don't know. Let me go back to this one. All right, with sharing. Go on, back you go, back. So this is the paper, a non-canonical tricarboxylic acid cycle underlies cellular identity. And here we look here. Shall I go through the non-canonical cycle again?
Yes.
Yeah. So you get glucose converted to pyruvate in the cytosol, shoves it in the mitochondria, and you get acetyl-CoA, which is then converted to citrate, and then swapped with malate in the cytosol through the citrate carrier or tricarboxylate carrier or SLC solute carrier 25A1. That's the gene. That then is converted with ACLY to acetyl-CoA and oxaloacetate, which is then converted to malate, swapped back with citrate back into the mitochondria. So this is a route whereby you have the glycolysis, but you don't end up with lactate. You instead end up with acetyl-CoA in the cytosol. Right? And going back to the paper, that's the wrong one. Sorry, let me just.
What activates the non-canonical Krebs cycle?
Well, this is where they don't have an answer on that, and I suspect, but it's a hypothesis, rather than something that is actually proven. Is it links in some way to low oxygen levels?
But it's just always going on. Is that right or no?
Well, in this instance, with the stem cells, yes, because if we go back to these stem cell niches being hypoxic thing, right, the stem cells are kept in a hypoxic environment. And obviously the original pluripotent stem cells that are in the egg that formed all of us is in a hypoxic environment as well.
Yeah, I never realized that. The body obviously maintains oxygen levels. It's very important. I didn't understand that stem cells and non-stem cells were kept at different concentrations of oxygen. I'm not
Yeah, this is the
sure.
first time hearing of that as well.
Well, that's why I thought it'd be all very interesting. But this is why, and I think it relates to this business about acetylating of the histone. So you're using the non-canonical TCA cycle to maintain higher levels of acetyl-CoA. Because, okay, this is where it relates to my hypothesis in terms of the substrate driving the long genes not transcribing. But it doesn't, it's all part of how does the body renew itself? Because the key issue about aging is how you get the body to renew itself successfully, which it doesn't do as people get older. But we know from cloning that that's not because there's anything wrong with the nucleus.
Yeah, I hate
When
to
you clone an old animal, you get a young animal.
Yeah.
gotta promote the conference, but it came up naturally this time. We're having a conference on June 22nd, and we have Shelley Buffenstein coming in. And just to remind everyone, Shelley Buffenstein is the researcher who discovered that naked mole rats don't age. So John said, yeah, you said non-Gompertz. That Gompertz just means exponential probability of death with the passage of time, increasing the probability of death. They actually don't age in the sense that there's not only non-Gompertz, but it's no aging. She published a paper, and now, these studies will take a long time. So there's only, I think, been one other that I know of, but it's been accepted that the probability of death of naked mole rats does not increase with the passage of time. And she discovered this by having this giant colony of naked mole rats where they mimicked the tunnels, but they were not, as far as I know, and we better emphasize this when she comes. I'll make sure to ask her that I do not believe they were kept in a hypoxic environment. They had
we
tunnels.
don't know, because if this is the tunnels, if they mimic the tunnels.
But they were above ground. They mimicked them in the facility, which is not underground, but they had tunnels.
No, it's a question where it would be nice to know what the oxygen levels were in the tunnels.
I'm not even sure. We'll have to ask her. I'm going to ask her.
You can ask her, because I'd like to know. But you see, have they measured the oxygen levels? Because obviously the oxygen levels would vary. They wouldn't be all hypoxic. They'd be going in and out of hypoxia.
But assuming they were not underground tunnels, they're above ground. So that means they're
certainly
not.
They enclosed in a box?
But it's not a molecular, an airtight enclosure.
Well, I don't know. We don't know, do we? But if
I'm
you've got
pretty
a tunnel, it's airtight apart from the tunnel, isn't it?
yeah, I guess you do
True.
need to measure
Yeah,
it.
so basically, this is the we know. I have again. I don't mind like doing a bit more research into the naked mole rats. I've focused
But
really on the whole issue about the HIF itself and HIF
if
one
I
alpha.
could show you that the naked mole rats in the non-hypoxic environment also don't age, what
Well,
would you
that'd be
say?
interesting. And then obviously that what that demonstrates is that there is another factor in their genetic makeup that prevents them from aging.
Yeah,
Yeah,
it's not
it
related to
doesn't mean
aging.
HIF is irrelevant. I think HIF is quite important. Uh,
It's well, I just want it. This is my perspective that the body has all these pathways that it could activate that are lead to longevity, but it's choosing not to. And you can trick the body, strenuous caloric restriction, strenuous exercise, a changing in the oxygen concentration does trick the body, and it messes with the body's active aging programs, and it does lead to some anti-aging effects. I'm not sure if hypoxia has ever done life extension, but it does regrow telomeres and
well, it's
stem
hard to
cells.
test, isn't it? It's very hard to test. So you could do it, obviously, but you've got to you'd have to do this varying of oxygen levels thing.
Yeah. But there's like caloric restriction, life extension,
Yeah,
exercise,
yeah,
life extension. I don't
yeah.
think anyone's ever done this regimen of hypoxia. I mean they say regrow stem cells or whatever, but it's how about actual life extension? I'm not sure we have
Yeah, well,
papers on that.
obviously, hyperbolic oxygen therapy has been used for human beings, and it has some beneficial effects.
Yeah,
Um,
a lot of people are doing it. I didn't know you were doing it as well, but you should know there's no life extension, right? You know that.
have they done? Have they done? My understanding from there were some experiments done in Israel, with HBOT, which found some benefits. I haven't seen anything that says HBOT does not produce life extension. But then you see, you've got to remember this point I make, which is too much of it is a bad thing. So too much hyperoxia is definitely a bad thing. You can do a lot of harm by having high partial pressures of oxygen for a long period of time. You have to keep it short and drop it back to normoxia or slight hypoxia.
So here's a question. When it comes to balancing the hyperoxia and hypoxia, um, there's a very fine balance to minimize risk
Yes.
of the hypoxia damage, ROS mediated.
Yeah.
What do you measure? What's the metric? What, not that
Well,
there's one golden answer, but just
yeah, but
what road do you go down to actually say that you have success? Do you measure outcomes? Are there any metabolites you can measure? Anything with
there are
more stable
metabolites.
half-life than ROS?
Yeah. The page johnhemming.blogspot.com hyperdoxia reducible factor links to a number of papers that have looked at specifically actually at the transcription factors. So the three transcription factors I refer to, they're all different papers from different labs, and they all do slightly different things. And in many ways, it's quite good because it shows how reproducible the response is, because you're getting lots of people doing it in lots of different ways and all getting a similar response, which is a little bit of a little bit of this process is a good thing, but too much oxygen is a bad thing, which is very much like hormesis in that sense. But yeah, I don't mind looking at the individual papers and talking about them, but it isn't something where it's something where there's actually quite a lot of literature which has tested it. What we probably need to do, you see, the problem is this: I'm doing all my weird biohacking type things where I do a lot of different things, including at the side of me, I also have hydrogen generator, which is intestinal gas. Are you aware of that?
Say to what
You know
gas?
what the
Say that
main
again.
gas is? Do you know what the main gas in farts is?
I do not.
Methane. Methane.
No, it's actually
Should be
hydrogen.
hydrogen.
It's
Ah.
Yeah.
hydrogen. All right. So, and then obviously hydrogen can get around. It's a very small molecule, as everyone knows, probably the smallest actual molecule with more than one atom in it. And it floats around the body, but it's very good for dealing with the OH oxygen species. So
Wait, what do you do with
Hypoxia.
hydrogen?
breathe it.
You breathe in concentrated hydrogen.
Well, it's not that concentrated. It's about 6%. Not very.
Well,
Not
what's the normal amount of hydrogen in the air?
well. It's not. It's nothing like 6%.
Okay.
Yeah,
You
trace,
get
right?
your intestines generate hydrogen,
Okay.
that's why it's the main gas in farts. I did find a paper which had analysed people's farts and concluded the main gas was hydrogen. Now
That's
That's
all for
there's another bit
science.
of news for you.
that's real science.
Yeah.
real science. Yeah. Well, I don't know. I probably could find. I can't find it in 5 minutes, but I can probably find it. Yeah, there is a paper that looked at what gases do you find, and methane's obviously there as well. But
Mhm.
the main gas it was found. And they did about 10 different people analysing their farms. And it's an interesting. It surprised me when I heard about it as well. And you can buy devices that separate hydrogen and oxygen in water, and then you can breathe it in. And I do that just to see what happens.
What happens?
It's difficult to tell because I look at the broader effects on my health, which I'm quite happy with. And the difficulty is when you do a lot of different things, sometimes you can work out the effect of any one intervention. Sometimes you've just got lots of interventions. So I do exercise as well, and various other things. And I can't say I have identified specifically what's caused by hydrogen.
Okay. But so what was like the thing that made you think to start doing it? Like what is it claimed by people that
Well,
what kind of benefit that it
there's a
has?
lot of people who argue a case for breathing hydrogen, and there's the various research papers. But you know, in one way, when it comes to interventions, I take rapamycin, for instance. I have taken rapamycin today, and what you do is you look at the risk and you say, well, what's the risk of taking it? And if there are arguments that may be a return, you might as well try various things to see what happens. And this is one of the great things of doing biohacking experimentation, which is you can try a range of things and see what the outcome is. And those things which you think seem to be working, you can keep going with. And those things which seem to be causing a problem, you can stop.
Okay. But so what outcome do people argue for that hydrogen will give you?
What do you want me to get you that one up? I can look up for it's on the internet. There are lots of arguments about it. It's it specifically says that you're dealing with the OH reactive oxygen species. So you've got the various reactive oxygen species that are created, and the hydroxyl radical is perhaps the hardest one to deal with. And when you have little bits of hydrogen floating around, and it gets all over the place, then that can deal with it. And your body naturally generates hydrogen anyway.
So basically it acts as like a chelation factor for
Well, it's sort of chelation, but it neutralizes the hydroxyl
neutralize, yeah.
radical.
Okay. Okay, so
The argument is it neutralizes the hydroxyl radical, but let me just let me look.
Is there generally an enzyme? Does that generally
No, it would just
require an
generally.
enzyme for
I
the?
don't think it requires an enzyme at all. I think it just goes in there and creates water.
yeah, because the
To split the H2 into pairing it with the hydroxyl radical, do you think that doesn't require an enzyme?
I don't think it requires. Well, I can look that up as well if you want. I don't mind. But
I don't think so.
Just since those radicals are so pervasive.
Yes,
if you've ever gone on a deep dive on what the mechanisms are of those radicals, I had one product that went into the depths on that, and just I'm not sure that we have that good of a grasp of exactly what drives them and exactly how they operate, just since they're so difficult to measure.
Yeah, well, they're very difficult to measure. So it's sort of this is one of the difficulties is that there are lots of Let me just stop the share on the screen and go onto a different share. So this is a paper here which was looking at the inhalation of 2.4% hydrogen, molecular hydrogen gas. All right. So there's quite a lot that goes on looking at using hydrogen. and they were looking at sort of safety here.
Mhm.
Let me sort. So, promising therapeutic agent for pulmonary disease of this argument here. Hydrogen water, are there health benefits? Right, and obviously hydrogen water is water with dissolved hydrogen. Well, you don't have to dissolve it; you can just breathe it.
Yeah, wouldn't it just like dissolve out of the water? Like, isn't it
Well,
gonna be unstable if you just put it in
well, I
there?
don't know. Again, I don't know the solubility of hydrogen in water and how quickly it come out. Obviously, if you know, there are lots of ways of dealing with hydrogen. Some people put magnesium in water and breathe it, and drink it rather. But
Mhm.
I hadn't this one here. Somebody saying benefits of inhaled hydrogen gas. Yeah, there are lots of therapies around that people use, and there, to me, seems to be no downside. You're breathing fart gas that doesn't smell, right? So it's a gas that's generated by your body,
Mhm.
which
Oh, that
arguably,
was my big question. Do you know with the intestinal production, is that human cell or non-human cell in the gut that's making the bulk of the hydrogen?
I don't know that whether it's the microbiome or whether it's the human cells that do it. I know it's generated there because that's where it comes from.
And is it through metabolism or
Yeah,
is
I
it
don't know.
known?
I'm sorry. I don't. I haven't studied beyond finding out that reading the paper which said that that's what fart gas is in the main. I haven't really tried to study why it's there. Sorry about
Well,
that.
I'm curious. I might try to take a deep dive and
Well, yeah,
report back.
it's not. Look, we started with oxygen. We're on hydrogen now. It's
We'll settle
Well,
you
until we
for
get to
the
argon.
entire.
well,
Yeah.
argon. We did talk about argon earlier. Obviously, that doesn't do anything.
I'm
Well, I
wondering.
assume it doesn't
Well,
do
yeah,
anything. I'd be very surprised if it did anything.
that's
I'm
the
wondering.
consensus. Yeah.
I'm wondering why the hydrogen is made by the intestines or the microbiota or like whatever is making, like what's the purpose of this and why is it in farts in the first place? Like if it's so beneficial, like
I don't know. We can try and find that out by doing some searching on it, but I haven't. What I've sort of like prepared for discussing oxygen, not so much hydrogen. You
yeah,
want to
no,
see
I
the
know.
hydrogen? You want to see the hydrogen machine?
Okay.
All right, we'll get you the hydrogen machine going. So this is a hydrogen machine down here. So that one is hydrogen. Whoops, there we go. And if we get, can you see the thing there? And that's then this comes up. Let me bring this one up here. Okay. And you have a cannula. I think it's a cannula or you have this thing anyway. And if I switch it on, so basically you can breathe hydrogen through it. Not when you're talking. And out of everything, you've got all these different. I've switched it off again now. It's just using hydrolysis to running a current through water and generating hydrogen and oxygen. There are lots of arguments about different ways of doing it, but I think it in the end comes down to this hydroxyl radical.
I see. Mhm.
Yeah, what's the relation between this hypoxia and your long genes theory?
I don't think there is a relation between them per se, save that. Clearly, because the hyperoxia inducible factor is a known transcription factor, and it has positive effects. My hypothesis is the reason long genes are not transcribed as a shortage of acetyl CoA. Now, there are two other hypotheses as to the cause of the non-transcription of long genes, and then there are people who would say long genes is not an issue. So I think they are separate issues. I don't think there is any reason why, I could be wrong about acetyl-CoA, yet HIF is still an important issue. You see what I mean?
I think so. Wait, say they're not directly related, is what you're saying.
They're not. Well, they are. Obviously, there's a connection because we're looking here. When you look at the non-canonical TCA cycle, you're looking at acetyl-CoA in the cytosol. And you're actually, when you look at the hypoxia that's involved in maintaining stem cells and the hypoxia that's required for conception, it appears, potentially, for successful conception. When you're looking at all of that, then clearly there's an interplay between all of these things. But then I don't have to be right about acetyl-CoA for there to be some relevance to HIF. HIF is just, it's a known thing. I just think the issue about hypoxia, the fact is, we're all people who've studied this issue to some extent. If it was news that stem cell niches tend to be hypoxic, that's a useful piece of news. If it's news that there is a non-canonical TCA cycle that I would argue is linked to hypoxia, probably, then that's a useful news. It's a useful area to look at because you're looking at a basic level of how all cells function. Because this whole point about transcription and the mitochondria is applicable to all mammals, and obviously to worms and everything as well.
Yeah, I think there are, an aging clock, and I think this goes into the theory. One explanation is the body's actively destroying itself, and you're interfering with those pathways.
Again, you could be interfering with those pathways by stimulating HIF to interfere with those pathways. So that's this is sort of perpendicular or orthogonal. It's not in the same dimension as the issue of the relevance of acetyl-CoA, even though it's connected.
Yeah, it's interesting because I think, caloric restriction also interferes with the aging pathways, but I think that is an evolutionarily conserved intervention in the sense that I think these pathways are designed to take cues from the environment and say, okay, this is the level of nutrients in the environment, how steady the supply of nutrients is, and we're going to age accordingly, just like your circadian clock is taking cues from the sunlight. I think your aging clock is taking cues from the availability of nutrients. Yeah, whether it's taking cues from the availability of oxygen or not, it could be interesting to think about. It's like it's a different type of intervention. You're messing with the aging pathways. I think that's clear. But is it? Yep.
The ROS to generate the ROS signaling. Well, this is clearly a response. If we look at those papers which we're linking to HIF, NRF2, and NF-kappa B, those are responses to ROS. Right? The more ROS, the more upscale it goes in its response. You start, as I said, with the building response. This is hormesis. It's very much whether hormesis always operates through this or not, or sometimes operates through this or not, I don't know. But it's the same sort of U-shaped curve type response, which is a little bit of stress causes a building response. A bit more response than you try and put the fires out with NRF2, and a lot more response causes harm. You bring in the army.
It's more than this. These hermetic effects, you can think of it like that, but then the question is, the body had the army. Why didn't it send in the army already? Why is it holding all these abilities in reserve?
Well, you need the army for something. So you need NF-kappa B under certain circumstances.
But there's no downside effects. Why do you have to trick the body?
Oh, there is a downside effect from having cytosolic ROS, because the cytosolic ROS can do a lot of harm to the cell. So it gives the cell ROS that it can use to attack something next to it. So this is partly things like warts and things like that. So, your body can fight warts better using NF-kappa B in the localized cells. It's not using the immune system to send in leukocytes.
Warts? Is that what you said?
It's not sending in the immune system. It's the cells have an individual ability to generate ROS and squirt them at other things nearby. That's your NF-kappa B, amongst other things.
Sure, but in general, your body, what you're calling the army, the body has abilities that it's holding in
It has
reserve.
these abilities at a cellular level, yes.
And it holds them in reserve and does not activate them until it's stressed.
That's right. And if there's a little bit of stress, it activates HIF. There's more stress, it activates NRF2.
Yeah, it holds them in reserve and activates these repair pathways in proportion to the amount of stress.
Well, NF-kappa B is not a repair pathway.
It is a what pathway?
It's a fighting pathway.
Sure, but it has beneficial effects more than just.
I don't know. I don't know. It's an interesting question. It's one, in many ways, the cells, different types of cells obviously respond in different ways, but I think they all.
I mean, when you work out, it's not just, your body responds by activating pathways. Not only do they repair your muscles, they make you much healthier.
Yes,
right?
that's right.
These are
making you much healthier thing, including, as I said, this, if you exercise one limb and the other one's in a cast, there will be a very slight growth, only a slight growth on the other limb. Which is that's sad I haven't got that reference to hand, because I'd like to give you that. That it's some things are more important to know than others. That in a sense is significant, because how it happens, whether it's through the transfer of mitochondria or whether it's some other sort of intracellular communication, I don't know. But it does happen. And obviously this is why the idea is, if you're doing exercise, it's good for your brain, because it probably does actually in some way affect your brain cells.
It's just, the question, I don't know exactly what I'm trying to ask, but we're interfering with the body's normal pathways and it has anti-aging effects. I'm interested in differentiating between interventions that evolution has prepared, is sought to deal with, and evolution is not prepared. I think caloric restriction, it knows, it's in our programming. If there's caloric restriction, we're going to respond in this way. So it has prepared to encounter this stress. Now, when you have too little or excess oxygen, it also, has a lot of pathways that get activated. I'm just trying to think, has evolution been designed to be prepared?
The extra oxygen will generate more ROS, because you're feeding that oxygen into the mitochondria at a rate that the automatic generation of antioxidants is not designed for. So it will create more ROS that are not actually not got rid of.
So it has more ROS than you think. Evolution has not designed us to be prepared.
It is an evolutionary response to a greater amount of ROS.
Yeah, then it activates the repair pathways that
it activates. The repair pathway is HIF1 alpha. NRF2 is not a repair pathway, nor is NF-kappa B. I talked to them about the builders, the firemen, and the army.
Okay, maybe also, can you talk? There's there's all different types of organisms. They live in all different types of environments with different concentrations of oxygen. There's no clear correlation between, you can point to naked mole rats, maybe they're hypoxic, but there's not a clear correlation between oxygen concentration and lifespan. And there are certainly organisms in the same oxygen concentration with different lifespans. So
well, obviously, yes. Mice and rats and human beings are all in similar oxygen concentrations. To me, oxygen concentration is an important thing that affects life more generally, and it's also a tool that can be used on a therapeutic basis. but it doesn't. You know, where do we come? We come back to this question of why do lifespans vary? I think part of it is mitochondrial efficiency and heteroplasmy and the development of heteroplasmy and the extent to which, for any one species, mitophagy occurs. So if a species has a better system of autophagy and maintains a higher standard of mitochondrial quality through genetic pathways, then that's where you're helped out, sort of thing. But we know, you know, as people get older, that you get more heteroplasmy and your mitochondria get less efficient. This is where caloric restriction helps by inhibiting mTOR and encouraging autophagy. And through the autophagy and the mitophagy, you're improving the quality of mitochondria by generally tending to kill off the ones with the worst membrane quality first, membrane potential. But that's again, you know, why is it that human beings have evolved a longer lifespan than, say, horses, which are quite big as well. And obviously that because of if human beings hadn't evolved it, we wouldn't reproduce adequately anyway, because we'd die off before reproducing adequately. But then, so there's more selection for better maintenance of the mitochondria, which, as you know, I think that's one of the two things that drives the shortage of acetyl-CoA issue, and also general inefficiency as well, because there's a secondary effect which is around the ribosome as well as the primary effect, which is transcription.
And how does this fit into caloric restriction? The
Well, caloric
hypoxia?
restriction encourages autophagy. This is the whole point about fasting.
And is that
you have this other question, which is whether, you know, and obviously if you've got higher glucose levels, you generate more ROS. And all of these things feed back, because as you lose control on glucose handling, then you generate even more ROS because your average fasting glucose is higher. So that generates more ROS.
I see. It's all about the mitochondrial damage. And so all these things are related.
Well, not all about them. Senescent cells have an important role in this as well. But mitochondrial damage is the point about the mitochondrial DNA, is it hasn't got a histone, so it's very vulnerable to damage that lasts. So once you've damaged the DNA for part of the electron transport chain, you're not, you know, yes, you might have multiple copies of mitochondrial DNA in the same mitochondrion, but once there's enough that produce duff proteins, you don't get the quality proteins. Now, hopefully, the damage is one that reduces the membrane potential, so the cell can get rid of the mitochondria. Because if it doesn't, because some things affect ATP synthase, and because they do that and reduce the ATP production without actually affecting the mitochondrial membrane potential, then the cell can't identify them for removal. That's a problem. But yeah, these are issues as to why certain things happen, why things deteriorate, and that we again, this is completely separate to questions of oxygen. Although actually, the problem with it, well, oxidative phosphorylation does involve oxygen, doesn't it? But yes, in a sense, everything's related, but you know, the inefficiency of the mitochondria is an issue.
Yeah, so my perspective is that the body, it has a clock and it's destroying itself. It wants to die right on time. And you know, that's why it's clear as excellent why do men and women have different lifespans, why do biologically very similar organisms have different lifespans, why is it holding things in reserve. I mean, it all I think points to this fact that the body's actually not trying its best to survive. And I agree with you, you know, the presence of oxygen was very important, and the body's delicate program has to take that into account. So when you change the concentration of oxygen, you might be able to throw off some of the programming. And it's interesting that it does hit stem cells, it does hit mitochondrial damage, so it hits maybe it hits telomeres, so it hits a lot of the hallmarks of aging. So it might
It may be
be hitting
useful,
this
yes.
program. Yeah, it
It
might
may be a
be
useful therapy.
affecting the program, but I think the real intervention should be with the design to attack the central program in mind.
Well, yeah, I'm interested in what proposals you have to deal with that. If I've got some things like breathing fart gas.
We gotta find the
Yeah.
clock. Yeah,
Sorry?
we have to find the clock.
If you again, obviously, I am open to being persuaded as to where this sort of scheduled clock is. As you know, I don't think that's how things work. But
I
I'm
think
very
it's in
interested.
the long genes, just like you're a big fan
Yeah,
of
it's
the
the long
long
genes.
genes.
Yes, it's we're not. If we both agree the problem is long genes not working, the only real reason is why the long genes don't work.
Did you know that the RNA transcript length change as you age? Do you know that?
Well, that's that. Well, this is the splicing issue, isn't it?
It
You
could
get
be.
shorter splices, yes. Because what it is, and this is, I think I did mention this when I was talking about this before, which is this question of what happens when you deacetylate the histone where RNA polymerase 2 is stalled. Does that create an alternative splice? Because that, you see, alternative splices have different transcript lengths.
Uh-huh.
And in fact, some things, some cancers, like prostate cancer, is particularly linked to alternative splices.
But alternative splicing, usually people talk about like there's one gene that can be cut up 3, 4 different ways. I'm
Yeah.
talking about a like a consistent, it's getting long. It's not like, that would a consistent. I can look at the length of your RNA transcripts and give you your age. So it's consistently changing. It's not like
Well,
it's
I'd be
just cut up.
interested in well, that's the long genes thing, isn't it?
It is disproportionately related to long genes. But what I'm saying is it could be somehow it's using that to keep track of time.
Yeah, again, I don't think that's how. I think it's just going wrong. I don't think it's doing anything which one would potentially describe, even in a Michael Levin type of sense.
Yeah, they just think it's going wrong. That's the main thing. We actually had a symposium here at Northwestern on Thursday, and it was like a lot of circadian people, and they just think no one believed in long-term clocks, so they just think it's going wrong. But if it's going wrong, how come, men and women age differently? Does that fit into your theory? It
Well,
really
yes,
feels like it's
because
programmed.
they have different genes and different, they are different in that sense. So,
I mean, there's certainly differences, but they're also very similar.
yes, I know.
And they have certainly the same oxygen concentration. So,
Yeah, I mean, obviously, it's not all about oxygen concentration. I just think that the normobaric oxygen paradox is a really interesting thing, and it gives a tool that can be used to stimulate the building transcription factor. So that's quite a good thing.
the paradox is that hyper,
The
uh,
paradox is the fact that it's the reduction in partial pressure of oxygen that stimulates it. That's the paradox.
yeah, anything that is, um, uh, so anything that's a deviation from the norm is
Well, it's
good.
not. Well, this is where you need to read all the individual papers that I've linked to and have a look at them. And there was one particular one, which, as I said, they used various levels of oxygen, including very low levels of oxygen, and they still got a response. And there's some complicated ways in which different things are created, and there's I think enzyme. There's a thing called FIH as well. But I think at the end of the day, focusing on the fact that you can do this is a good thing, because you then need to see what effects it has.
Yeah, John, you should consider coming to Chicago on June 22nd. We're going to have the Naked Mole Rat people there, and we're going to have the Long
Well, I
Gene
probably
people
have to.
there.
You're not doing remote stuff, are you?
I probably will do remote, but I want to get you there in person, but there will be a remote option.
Yeah, well, if there's a remote option, when is it?
June 22nd. That's a problem. Should I have a remote option? But once I say remote option, people just say I'll zoom in. But I want you there.
Yeah, well,
It's nice to have you could talk to these. You could contribute. I think you'd be a great contribution to the discussion.
yeah. Let me think. It is quite a commitment to fly to America from the UK. And I've got to look at what other commitments I've got around that time.
Yeah, it's a big deal, but I just announced the hotel and we're like 85 days away. So time to
Yeah,
start
no,
planning.
yeah. It's just so I obviously I can zoom in, but actually I should. Well, funny technically, I've got a jazz gig somewhere around about then. I'm booked to play somewhere.
Oh, I didn't know you played jazz.
Yeah, it's on my YouTube channel.
Oh, nice.
What's your YouTube channel again?
John Hemming.
How long do you want to live, John?
I haven't specified a period. I want to be healthy. I don't want. I want to live healthily. I don't. I'm not really too worried about how long it is so much. It's the. I haven't set a time. I haven't said I want to live for sort of 103.5 years or anything like that.
Assuming you're healthy.
I just want to improve my health. So, and my health at the moment is actually, as far as I'm concerned, really quite good. So I'm quite happy. So but I'm doing, as you know, I do these weekly blood tests and on seeing where it all goes.
Yeah, we're learning a lot. We knew about the blood test. I didn't know about the concentrated oxygen and the hydrogen. And
Well, yeah, you don't know that you can buy machines to make fart gas, do you?
Yeah, I did not know.
John, who are the agony ants?
Sorry?
I found your YouTube channel, and you have a lot of videos that are titled Agony Ants and the Journalists.
I don't. Oh, well, I've got loads of things. There's the agony aunt and the journalist. I'm
Yeah,
sorry.
there's
That's probably sometime, probably when I was an MP or something like that. But
yes.
I can find you. I can find you a jazz video if you want. All
No,
right?
I found them. No, I'm just like I'm scrolling through your catalog right now. You
Yeah.
were an early adopter to YouTube.
Yes, I was. Yes.
Wow. Your first
I
video is from 17 years ago.
Oh,
yes.
wow.
I was very early on the internet. In fact, I set up the first e-commerce business outside America.
That's
And
awesome.
I put the first bank on the internet and various things like that. I've been in
Nice.
tech for a long time. I set up a tech business in 1983.
hmm Did you ever like have this point in the internet where you knew people's names by their IP address or vice versa?
No, I didn't actually, because that was, yes, okay. Well, it was still managed. We were still managed on IP version 4 anyway. We haven't had to go to IP. I know people are generally using IP version 6, but IP version 4, we haven't actually run out of numbers yet.
See. Yeah, because my teacher for a class I'm taking right now, he said that I think it was his father-in-law was like a super early adopter, like back for ARPANET. And so
Yeah,
he was the 14th person on the internet.
quite possibly. Yes, I was 1985. It's 1995 rather. Sorry, I was actually 1994. I think late 94.
Oh, okay. Oh, so when I was 4 years old.
Well, I don't know, but you do.
I was.
Yes. Well, I was 34 years old.
That means that you're 30 years older than me.
That does mean I'm 30 years older than you. Yeah.
Sorry,
I had
we
my
were talking about.
I had my 64th birthday a few weeks ago.
Oh, happy birthday.
Thank you.
Oh, 64. Happy birthday. Yeah.
Yeah.
Just like that song. When I'm
64, yeah,
Yeah.
when I get older, losing my hair. Well, it's sort of growing back slowly.
Nice.
Many years from now.
Did you play that song on your birthday?
No.
No.
I was in the St. Patrick's Day parade in Birmingham the next day, but I was playing the guitar and it was singing A Long Way to Tipperary.
I don't know that one.
A long way to Tipperary, it's a long way to go, etc. Because it was the Tipperary County Association.
Uh, what's
I see.
Tipperary?
Tipperary is a county in Ireland.
Um, it does sound like a vaguely Irish name.
Yeah, Hemming isn't. Hemming is an Anglo-Saxon Viking name. It's a Viking.
We
Yeah.
had in Chicago, we had a huge St. Patty's. I didn't know
Yeah.
Chicago's known for St. Patty's. We dyed the
No,
whole
but
river.
Birmingham is very big on that. Yeah.
Oh.
And I have a couple of ancestors who were born in Ireland, but mainly, as in my family name is Viking. But it's been in the UK for like ever since we invaded in about the year 800.
Nice.
Same, relatable.
So, like, one of the things I'm trying to do with this conference is to promote this just to a wider public. You must know that most people in studying biology and academia are not interested in aging. They don't think of aging as a disease. So I would like to
Well,
think,
I think what the issue is to what extent people recognize that you can do things on a practical basis that change the trajectory. And then there is this question, the underlying question is one of to what extent you can fix the long genes issue, right? And I think you can, but I think it's a matter of persuading people. And I think once you persuade people, then people's views will shift on it. But I think
there's been an incredible dearth of research. Yeah, the extent in which we can intervene in the aging process. That's what I would like, people to realize. Yeah,
yes,
exactly.
well, this the problem is it's aging research, as in the search for the fountain of youth, um, has gone on for a long, long time, and
As it
actually
should.
and yeah, and only more recently and the geroscience hypothesis, I think has, I think there's no question that's proven
What
that
is that?
the geroscience hypothesis is if you intervene on the basic points of aging, you fix a lot of things at the same time.
Oh yeah, I agree with that 100%. A lot of people would
Yeah.
disagree with that though.
Yeah, well, we need to have a debate with some people who disagree on it. There's no sense us having a debate when we all agree. We don't get anywhere with that. The interesting thing to me on that, for instance, is that if you look at things like AMPK activation, actually in traditional Chinese medicine, there are a group of herbs called Yang-Qi activators or Yang-Qi, and they're all AMPK activators at the same time. Things like ginseng and ginkgo biloba, they happen to be all AMPK activated, which is an interesting thing. And there are other things, HDAC inhibitors that are polyphenols tend to be HDAC inhibitors. And so that you have things that are part of traditional medicine that actually act on the aging pathways. They may not act sufficiently to make a massive difference. I don't know. Yeah, one of the things I do, for instance, is when I'm doing AMPK activation, I take a large number of activators at the same time. So I take about 14 different AMPK activators, which I have a list of somewhere. I've got them all in a box.
Yeah, so you have a lot of interventions on a lot
I
of
have.
different fronts.
That's right. Yes, because I don't think you will find a single intervention that deals with all the issues in an ideal way.
And you take these weekly blood tests and you're convinced by the blood tests that your interventions are working.
Yes, I'm convinced by the blood tests and other results. So I, for instance, have continuous glucose monitoring and other things. I
What
think they are.
exactly in the blood test is most convincing to you?
Well, C-reactive protein is perhaps my top priority. And then you see, the thing is, you get things like ferritin. Ferritin, I've had some really quite good results in. But with biomarkers, biomarkers are complicated because if you take a biomarker like creatinine, it metabolizes in blood and it's affected by muscle mass. So as you improve your muscle mass, then creatinine goes up. So it can get a bit confusing. I don't know to what extent you followed Morgan Levine's PhenoAge spreadsheet. Yeah,
I've read the paper. Yeah, some of it. Sorry.
One of the figures in there is creatinine, and there are lots of problems with creatinine. It's a very unreliable biomarker.
Oh, I agree 100%. Yeah, I hate to make fun of anyone's aging clock, but a lot of those biomarkers were unreliable,
No,
and they were just
actually, no. Morgan Levine's work on that is quite good. It's based on biomarkers that themselves are not that reliable.
So this is something I've been wondering, how much stock do you place in telomeres as a useful biomarker?
I don't think they're that useful because I think they arise from how healthy cells are when they're replicating. And we're not actually encountering a replication problem. We don't have, I don't think we're saying that, the body's failing because it can't replicate. Some cells hang around for a long, long time. What we're having is the certain cells are not being created. And that's not because they can't replicate, it's because they can't differentiate. It's an interesting measure, but I don't worry about it.
Okay. Yeah, that was my impression as well, was that like telomeres used to be held up as like the big deal of like the biomarker of aging, but it seems like pretty much no one who is working with clocks, like epigenetic or otherwise, seems to be really concerned with them. And
yeah, I don't think you've got telomerase in your genome, and basically the question is what switches it on when it needs to switch on, and but are we? Yeah, Hayflick's limit, fair enough, okay, but that was in vitro cells, okay. So are we encountering a problem that you can identify caused by the shortness of telomeres? And I don't think you can say that.
yeah.
We haven't identified what the problem is, and therefore solving that problem however you wish to do it. People have a tendency to look for a silver bullet. They tend to look for a single solution that does everything. And I don't think there is going to be a single solution that does everything. I think it's a combination of interventions that will improve various things.
And hopefully can be put into a single pill or injection.
I don't think you can do that. Yeah, people would like that, but I don't think that's going to work.
There is a single thing that cures aging. It's having birth. The babies are born young. We just need to figure out how to trick the
Figure
body.
out why it is. Well, we know a lot of these things already. We know it starts with hypoxia.
Yeah, I was curious to see what you were saying about that. Okay, I mean, it is true that it's less oxygen in embryo development than, after embryo development. What were you saying there?
Well, only really that it has a relevance to pluripotency. And I really was in a sense skirting around various things before I wanted to go into
Embryo development is very, controlled, right? They have to have that controlled environment of exactly the right acidity, the oxygen concentration.
Yeah.
That is more
but
just about.
It doesn't get us anywhere really, because I don't think, at the end of the day, we are sitting around and we're all aging at a particular rate or whatever. And the air oxygen concentration where you are is probably much the same as where I am.
Yeah, I'm just not convinced the oxygen concentration is that important.
I think it is important if you want to stimulate HIF. But it may be that if people live at say about 17%, they live longer. I don't know.
Wait, and you said that it was also activated during exercise, right?
Yes, HIF is well, you get mild hypoxia, don't you?
So exercising is much more beneficial than, I don't know, some concentrated oxygen mask.
Well, people are using HBOT for various things, right? I think the mechanism for HBOT is HIF. People are paying for it. That doesn't mean it necessarily works, but there are, and obviously there are papers that say it does have a positive effect.
So we could test it by like seeing if you do the oxygen like mask thingy independently and exercise independently, and then like measuring the effects of that, and then combining the two things together if you
Yeah.
see an additive.
There are lots of ways you can do things, and there are people who do exercise with oxygen concentration. Now that's a real fact because you have to do your exercise with a mask on and breathe this air. And it's like I sit here in the morning and I attach my hydrogen for a little bit, and then I attach the oxygen for a little bit. But whilst I'm doing that, I'm doing other work on my computer. I'm not sort of having to focus on gas therapies.
You do this every day?
I do, yes, pretty well. I'm going to leave Birmingham for a few days soon, and I will not do it whilst I'm away from Birmingham. But you see, the underlying problem is I've got a group of therapies that are, in my view, quite beneficial. But I know what the individual effects of some are because you can identify them. But with a lot of them, I don't know. So I keep going with everything, and I may at some future stage try identifying the individual effects of one. But at the moment, it's a very slow process. The process of change is not rapid because you first have to get the cellular environment to be correct. And once the cellular environment functions, then it takes a while for the cells to grow differently.
What is your opinion of Brian Johnson?
I don't think he does that much on. He's a bit more of a magpie than I am.
What do you mean by that?
He
blood
gets all the
transfusions.
shiny bits and has a go at them.
You see.
Wait, what does that
I
mean?
personally don't think using Botox is an anti-aging technique. I think using Botox is a cosmetic change, which, if you actually manage any anti-aging in what you're doing, will be a long-term nuisance because you've killed off some of your cells that you probably would prefer in the long term not to have killed off. So I'm not a fan of using cosmetic intervention simply because it conceals what's actually going on. Because your underlying problem is whether you're improving your overall health. And when you start killing off bits of your body so that they don't look wrinkled.
He does a lot of stuff. Is the cosmetic stuff a big part of what he does?
No, he does a lot of stuff, and some of it's the same as me. So I do some of the things he doesn't do everything that I do.
He does the blood transfusions. Do you do that?
He does. Yeah, I don't do blood transfusions, but I do give away. I obviously give quite a bit of blood in blood tests, but it's not that much.
Yeah, you need a lot more than that. You should just start donating the blood. That honestly, the blood, getting rid of old blood is probably much better than all the oxygen stuff.
Well, remembering that I do a blood test every week, that's 10 to 20 milliliters a week. And ferritin is the measurement. My ferritin levels have come down from about 3 to 400 to about 80.
Yeah, actually, that's probably very good, actually. Giving
That
the blood.
that is ferritin. That movement in ferritin is really good. The CRP, my CRP is normally under the measurable threshold of 0.15 milligrams per liter, which is lower than Brian Johnson.
So you're doing better than him, it seems.
On that measure, not on all measures.
I see. Yeah, we were digging into the convoys and the like heterochronic parabiosis whole thing like a few months ago. And what I learned is that the beneficial effects of heterochronic parabiosis and like the idea behind blood transfusions in general seems to be more mediated by plasma dilution versus anything else.
Yeah, well, there are two issues in this. I think one is the extracellular vesicles, and there is, there's evidence that obviously when you do exercise, your cells will spread around mitochondria to try and provide better mitochondria where they're needed. And the other one is interleukin 10, which clearly has its effects. And when you do plasma dilution, you're going to take out some interleukin 10. I
What
haven't done
is
plasma dilution.
is
Haven't tried.
interleukin 10 good or bad?
Well, the interesting thing about interleukin 10 is it does cause senescence, but is thought to be anti-inflammatory. That's because it inhibits NF-kappa B through the Janus kinase. But I think it's one of the drivers of extending senescence. It causes patchy senescence.
Oh, so it like some cells will become senescent in a given niche, but some others will not.
Yeah, it's all these things are all probabilistic to a certain extent because it depends on how much of it hits any particular cell. But there, as I said, there's this paper in Portugal that where they did some experiments with interleukin 10 and found that it would cause senescence in
Hmm.
vitro.
Well, that's not good.
Yeah, well, it's interleukin 10 is part of SASP. So if you've got part of SASP that you know causes senescence, there is an issue there. And what you find is that that senescence is patchy. It tends to spread.
Right. Yeah, yeah, that sounds like what I'm familiar with it as well. But
Yeah, well, in all sorts of, like kidney disease, patchy senescence in the kidneys. Now, here's an issue for your uh, naked mole rats, which they apparently don't have a lot of senescence, which is interesting. Don't know why, but
Every organism, there's no universal hallmarks of aging. Not every organism has senescent cells.
yeah.
Yeah. There's a lot of hallmarks of aging that other organisms have that we don't have. So
Mm.
you could also ask like the nematodes, their mitochondrial fractures in a very characteristic way.
Mm.
Why doesn't that happen to humans? The yeast
Well, because if it
accumulate.
did happen, we probably wouldn't live that long. And so we would, if we had mitochondria as bad as the nematodes' mitochondria, we wouldn't survive to reproduce.
Hey, do you know this is one of my favorite studies? Is the nematodes have the Dower state. Have I told you about this? That they go into if they have like four larval stage, four puberties, and if you trick them into thinking it's an adverse environment like overcrowding or heating or something, before the fourth larval stage, they go into a Dower state, this
All right.
diapause state,
Yeah.
and they usually live like 2 weeks, but they can last months in the
Yeah.
Dower diapause state. And so one of their hallmarks of aging is the mitochondrial fracturing. And it happens in the Dower state too, even though it's slower. And then if you remove the adversity, like give them enough food or remove the population density, then they exit the Dower state and they live their normal life course.
Yeah.
And
Which is
what happens?
interesting.
Yeah,
Yeah.
what happens to the mitochondria? All the fracturing disappears. So, some evidence that whatever you consider an age-associated change with the mitochondria, the body actually already has the ability to repair that so-called damage and revert the organism's mitochondria back to a youthful state. It's all about the clock. Something is timing it, sending the signals that, to go back to the youthful state.
Hmm. Yeah. Well. Yeah. We can. This is where you get this interesting question again about development, which is obviously we have the metabolic shift from a hypoxic environment through differentiation to a normoxic environment, which then maintains differentiation at that position, possibly through the non-canonical TCA cycle. We don't know. And then you have gradual deterioration in mitochondrial quality, the development of senescent cells, and its consequential effect on the transcription of longer genes and translation up to a point. But we don't know.
Yeah, I mean, I'm interested in hypoxia in the sense that I think it can perhaps interfere and perhaps affect
But you
the
can
sick.
interfere, yeah. I just think it's a potential tool, or one of a number may need to be used. But if it's a tool, if you're talking about trying to avoid diabetes, it's worthwhile trying to do that.
Hmm.
For instance, just giving one example.
Yeah, and I was also going to ask you about, so since you have all the experience in like politics, so if I wanted to promote aging research, particularly the idea that death is not inevitable and there's a lot of interventions that society should prioritize. Yeah, what's the best way to do that?
Well, there is in Congress, as I understand, a congressional group which is interested in that particular subject.
Alliance for Longevity.
I don't know what they're called.
That's what they're called.
I should know this.
Is that a congressional group?
Yes.
Right.
It's organized by this guy, Dylan something, who I actually met at a conference.
Yeah.
Yeah, and I believe Newt Gingrich is on it. But yeah, like he intentionally went for both like Republicans and
Yeah.
Democrats.
Well, that's the thing about it. You're best off going. You don't try and make it party political because it doesn't.
Yeah, of course.
You what you're trying to do is get into the machinery of government. Now, the thing about organizations like that is they include Congress people who will know who to talk to. And so it's often a better mechanism for that's an international one. That's international.
Is this what you guys are talking about? Oh no.
No, there
No.
will be something in Congress. There will be a group in Congress. I saw
Yeah.
it had been created. They all parliamentary systems tend to have such groups. Do you want me to try and find out what it's called?
Yeah, I should know about this.
Let me find out.
Yeah, because in I believe in the UK they have
Yeah, there is one in the UK, yes.
yeah. You guys have a different like fancy name for it than we have
All party
here.
parliamentary group.
Okay. And this is the
Now,
just put in the this is the one that I'm talking
now
about.
there is I have found reference to it, but I haven't found the name of it. So I found it on the Methuselah Foundation, New Congressional Caucus. So it's called yes, the Congressional Caucus for Longevity Science. That's what it is. Yeah.
Yeah. Okay.
So let me just see if I can find anything else on this.
Oh, so is that the caucus you're talking about? Is that the
Yeah.
same as okay.
So there's a thing called on Lifespan IO have a reference to it as well.
All right.
It's a congressional caucus for longevity science. So that's what it is. Yeah.
Sky. Yeah, Dylan. Yeah, he's who I met at the Buck.
Yeah. Well, that's a different thing. What you want is you want to work with the congressional. If you're working in the U.S. jurisdiction, you want to work with the congressional caucus because they're in the system rather than outside it.
Oh, Irina's there.
Yeah.
None of these guys believe in programmed aging.
Yeah, but this
Just-
isn't the people you need to. These are not the ones I'm suggesting that you need to deal with.
Oh, who were you
Yeah.
suggesting?
Sorry,
The
yeah, I
congressional.
spaced out.
All right, let me just give you a link to a story about it. It's a story on Lifespan.
Oh,
In fact,
Sam,
shall I share?
fantastic.
Shall I share this to you?
Yes.
If you unshare what you're doing, I can share screen. Yes, I can share screening. Bing, 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 bing. This thing here. All right, announcing the formation of the Longevity Science Caucus. That one is your group. All right.
Well,
Oh,
this is the
actually,
same thing.
no, it isn't because
Okay.
it's the Alliance for Longevity Initiatives would like to applaud the efforts of Congressman blah 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 for co-chairing and launching the newly formed bipartisan Congressional Caucus for Longevity Science.
You're right.
Now,
Wait, it's a different thing than we were looking?
maybe linked to it in some way, but actually you want to see the longevity science. This is the thing. All right. So you've got a republic. You should always do these things on a non-partisan basis. You're more likely to get results. And
Oh,
basically that's the press release.
Anna Eshoo. I don't remember if I voted for her or not.
Is she in your particular area or not, or what?
Yeah, I'm in California. I remember like we had an election a few months back, and I saw a whole bunch of billboards campaigning for her. I
Well,
don't
anyway,
remember.
that's so that you've got that they are a longevity science caucus, and that's going to be a good mechanism to make contact within Congress.
So what's the difference between a caucus and just the like whatever the Alliance for Longevity is
Well,
then?
the Alliance for Longevity is not a body particularly associated with the US Congress. The Alliance for Longevity, as I understand it, is an international lobby group for longevity issues. And what your question was, how best to influence US government policy. And the best way of influencing is working with Congress people to do that. And you want ones who are interested, and these people are interested.
Okay. I don't think that Alliance for Longevity is international. I think it's pretty specifically America.
Is it? I don't know. The one you linked to was international, but Alliance for Longevity Initiative. Let me have a look at this. Alliance for it's a non-profit. It's an American non-profit. Yes. Um, they're obviously liaising with the caucus as well, which is sensible. About who we are. And they obviously helped form the longevity science caucus, they say there, yes. Maybe
Yeah.
they are just American, I don't know. There are quite a few different groups.
I've only heard of these two now, but yeah, I guess there is a lot of like non-profits like Methuselah and whatnot, but I guess we
Yeah,
were
yes, there are lots of
yeah.
those.
Yeah, that's true.
But this, you
Oh
see,
yeah. The right to try things. I remember like this was a big deal because it was either it was like there was some law passed saying that like you cannot just take rapamycin willy-nilly, like it has to be more regulated or something. And that made things more complicated. So yeah, the right to try was important to these people.
yeah. Well, I do think that these are important arguments to be had, which is sometimes what you find is that people in the Department of Health tend to think people should only do what we think is right.
Yeah.
And that's dangerous because they're not always right. But they tend not to think they may be wrong. And there's also people's freedom to make stupid decisions. You
It's
can't.
true.
Yeah,
So I got mixed feelings. I think, the freedom not to, try to live forever. That's a freedom to make stupid decisions. They shouldn't have that.
well,
But
people. The difficulty you've got is if you limit people's rights to make decisions which you think are wrong for. The danger is that somebody somewhere is making decisions as to what should happen to you that you are wrong, and in fact they are wrong as well. So it's sort of people need a certain amount of freedom to do things which are stupid for themselves,
yeah, not
as long as it
because
doesn't affect
it's
others. Sorry?
only because, if they don't, the power might be abused by
Well,
someone.
no, the power is abused. It not just might be abused; it is abused.
But in general, if we all agree that this thing is stupid and no one's, even the person doing it's not in his self-interest, and if he was smarter, he wouldn't make this mistake,
Well,
then
what
we
about
agree
drinking alcohol then? What about drinking alcohol
yeah,
or
I'm against it.
smoking tobacco
I'm
or
against it. It's not in their
smoking
best
weed?
interest.
How about smoking weed?
I'm against it all.
Yeah, well, that's your personal view.
But it's objectively bad for these people. They're not making a
I
good
no, no,
difference.
look, it's one of these things. We probably take different views on these things. I think people have a right to do things that are harmful. So they can. I used to be obese, as you know, and I changed that through my own decision to change it, not by external factors.
No, you learned from it and you gained a new appreciation, I would assume, for
Well, yeah, but I decided it was a sensible thing to be smaller.
yeah.
Be smaller, but I still drink alcohol.
Yeah, it's like with stuff like that, the poison is in the dosage.
Wait, you drink
Um,
alcohol, John? Even though you do all these interventions to live healthier, you still do something that you know
Well,
is not
I
healthy.
think I thought you knew I drink alcohol, but yes, intermittently, not always. I'm not drinking it today.
But
I've
you,
drunk it, but it went on calls.
yeah, I remember that.
Yeah, so
I
but
think I
I also
remember.
take pantethine dihydromoracetin and intermittent molybdenum as well. Molybdenum obviously increases aldehyde oxidase. Pantethine is a non-rate limited version of B5, which creates the coenzyme A precursor. And that accelerates the conversion of acetaldehyde into acetate. And acetate is mildly helpful. So you have ethanol, which is neither here nor there. It makes you woozy and isn't particularly good. It creates acetate, but that's bad news. That causes all sorts of problems. But if you can get rid of that, it doesn't do you so much harm.
Well, that's good to know.
These issues, people go too far in one way. Like, obviously, if a child is going to kill himself, we should stop him, right? It's not in his interest, right? He's not making a rational decision. And honestly, I feel the same way about a lot of alcohol and drug use. It's not in their best interest, and we do need to stop it.
Yeah, but there are two separate questions there. And one of them is what you achieve when you try, which isn't necessarily
It
that
is,
good.
it's tough if like the entire country wants to do it, and then, we make a law against it. We're going to war with these people, and then there's casualties in the war. Yeah, I don't know what
Yeah,
to think about that.
prohibition was tried in the past. But what I would suggest is that we consider philosophical discussions as to how much freedom people should have to take stupid decisions probably is not in order for the conversation about hypoxia.
Oh yeah. Or we're talking about like getting people to prioritize living longer. I think they're making a wrong decision. And you know,
Yeah, but let people make wrong decisions. You know, well, it's one of these things, isn't it? It's sort of let me stop the sharing anyway. I'm not puritanical.
Me neither. So I
I
agree.
guess I'm a little more puritan. I know
No,
what's right.
you have a right to be puritanical. I have no objection to you being puritanical, and you can control yourself as much as you want. Just don't control me.
And I know what's good for the people.
Yeah, I have some idea of what's good for people as well, but let's not debate it here. I don't think we're. Yeah, it's not really within the context of this.
Yeah, but no, thanks for preparing. The hypoxia, very interesting.
Well, hopefully useful. And obviously, if you could get me the recording, then I'll put it up on YouTube. And we had about 100 people look at the start of the last video. I Who knows? The hypoxia is very interesting. There's lots of interesting stuff. But thank you for agreeing to let me talk to you again.
Yeah, no, it was a pleasure to have you. And yeah, next week we have Sam, you're speaking, is that right?
Sam's there, but
He's
not
there,
there.
but he's on mute.
He's on mute.
So he
Yes, I had to navigate to the phone. Yes, that is right.
Okay, so we got,
Okey-dokey.
and you wanted to do Sunday next week, is that right?
Yes, that's correct.
So
Yeah,
we'll
I
do,
may not be able to attend on Sunday. I might be able to attend, but most likely I won't be able to make Sundays.
yeah, sorry for mixing up the time. I think we're gonna start doing consistent. That's okay with like Jackson and everyone. We'll try to do so actually right now it's 1 p.m. for me. This is the usual time. We'll do 1 p.m. Saturday, but if the speaker wants to switch to 1 p.m. Sunday, we'll switch. I think that will
Okay,
be it. It'll be
that
more
should work.
consistent for everyone.
But what is probably best is to agree the day and the time a couple of weeks out.
Yeah, at least a week
Very
out we need to
interesting.
do. Yeah, I
Right.
agree with that.
There
So,
are
okay,
other people interested, but you can't advertise it with if you're going to move the time.
yeah, that's
Yeah.
fair. So let's decide, we should decide next week at the current meeting. So next week it'll be this time, 1 p.m.
Yeah.
On Sunday though.
Yeah, apologies from me, but I won't be able to make it on Sunday.
And that's okay. Yeah, we'll switch back to Saturday, so we'll
Yeah,
get you
and then I will
next
be
Saturday.
able to. Yeah. All right.
Okay. All right.
Nice
Yeah. So
talking to you all. Anyway.
yeah, thank you everyone for coming, and yeah, thanks for the presentation.
Okay. No
All
problem.
right. See you guys next week.
Bye bye.
See you then.